Hi, I'm Alex Fong, Vice President of Engineering at Hinalea Imaging TrueTech Technologies, and I'll be discussing our work in the detection of foodborne pathogens using darkfield hyperspectral microscopy. Foodborne pathogens such as Salmonella, Campylobacter, and E. coli are a significant threat to public health. The impact in the United States alone is estimated to be over $70 billion a year. Conventional detection methods depend on biochemical assays and are time-consuming, and that adds to the cost and severity of outbreaks. Hyperspectral imaging technologies show promise in addressing these issues. Such methods allow specimen samples to be extracted during processing and even possibly as part of inline inspection of the product itself. Current methods, which include plating, molecular detection, and microscope-based non-imaging FTIR spectroscopy, involve multiple manual steps from sample preparation to incubation to analysis. The time typically to answer ranges from 18 to 60 hours. Hinalea Imaging has developed a hyperspectral imaging microscope peripheral based on the Fabry-Pro variable etalon interferometer, the Model 4200M. Unlike most hyperspectral systems, which are based on line scanning or push-broom grading technologies, it does not require physical scanning of the sensor. The interferometer is placed in front of the sensor, which functions as a tunable filter that can sequentially select spectral bands and generates the hyperspectral data cube by collecting complete images at each spectral bandpass. In darkfield microscopy, a center field stop at the illumination source allows only light scattered from a specimen to enter the objective lens, so cells appear as bright outlines. Hyperspectral imaging of specimens using this method can yield unique spectral signatures, which in turn can be used to identify pathogens without the need for staining or special reagent growth media. Hyperspectral imagers generate data cubes which contain spectral information for every pixel in the image. Machine learning algorithms are needed to interpret such massive amounts of data. Typically, they are used to group elements in an image by common spectral signatures and spatial characteristics such as cell morphology and image intensity. Training them on large sample populations enables us to optimize their performance. This approach allows us to turn such images into actionable results. In collaboration with our industry partners and the USDA, we've been able to test the feasibility of identification of foodborne pathogens utilizing dark-field microscopy and hyperspectral imaging. We've looked at bacterial organisms such as Salmonella, E. coli, and Listeria and collected data cubes of numerous and various isolated and mixed bacteria, analyzed the spectral spatial profiles of the sample, and demonstrated the ability to identify pathogens, even in mixed samples. Our initial studies utilized the Model 4200 coupled to an Olympus BX43 microscope equipped with a dark field illumination source. The IR filter was removed in order to capture the entire visible through NIR spectral range within the response of the imager. The classification methodology involved the following steps: identification of cells based on the sample image using OpenCV library tools, calculation of the mean spectra of each cell to create a spectral reference library, use of the spectral angle mapping algorithm, calculation of a spectral angle between the library spectra and the mean spectra of each cell, setting an appropriate threshold for inclusion within a class, marking the cells within that threshold as positive using a green colored outline and otherwise as negative using a red colored outline. This image shows the OpenCV tools being used to identify the E. coli cells within a field of view. The bright red plot in this spectral profile represents the average spectra for all the E. coli cells in the previous slide. The same process was followed with the Listeria specimens. These are Listeria cells identified by the OpenCV tools. And this is the average spectra for all those cells. Comparing the two specimen spectral averages, we can identify distinct differences. The Listeria profile is narrower and centered more towards the green end of the visible spectra, whereas the E. coli profile is wider and centered towards the red range. We then captured hyperspectral data cubes of a mixed specimen slide of E. coli and Listeria and classified them using the two spectral libraries. On the left, the E. coli spectral library, on the right, the Listeria spectral library. The green positively marked cells on the left and red negatively marked cells on the right show excellent agreement, and vice versa when compared. The USDA ARS data included an expanded set of pathogens. A Nikon E80i microscope and halide light source were used. The data was classified not only on the basis of spectral profiles, but also morphology or shape, and intensity images or distribution of brightness. To do this, a hybrid deep learning framework was developed. In addition to the spectral averages used as reference libraries, intensity images which were normalized were used. Scalar normalization was applied to the morphological, and an automated optimization method was employed for automatic tuning of all the parameters and factors involved. The performance of this approach was compared against those from the conventional approaches previously discussed for 5 common foodborne bacterial pathogens: Campylobacter, E. coli, Listeria, Staphylococcus, and Salmonella. The deep learning approach scored significantly higher than the conventional techniques. Principal component analysis, or PCA, was used to simplify complexity in the high-dimensional data while retaining trends and patterns. It transforms the data into fewer dimensions to summarize features by geometrically projecting onto lower dimensions, and finds the best summary of the data. Plotting these results demonstrates the ability to separate the observed pathogens into distinct groups. In summary, we have demonstrated the potential for dark-field hyperspectral microscopy to be used for pathogen detection in specimen samples drawn from food processing operations. In collaboration with the USDA ARS, we observed higher performance through the use of hybrid deep learning frameworks. The approach requires almost no preparation of the sample and has high sensitivity and specificity, as it can detect and identify a pathogen at the single cell level. And unlike biochemical assay techniques, there is no requirement for some critical volume of cell material needed to react with an analyte. The enrichment was only used to facilitate the location of the cells within the slide's field of view, and there is potential to shorten the time frame required and possibly eliminate it completely. Within the framework of our collaborative research between Hinalea Imaging and the USDA ARS in Athens, Georgia, we plan to continue to look at other classification algorithms to improve performance. We also intend to explore other light sources and integrate an SCMOS sensor in place of the current CMOS device for higher sensitivity and shorter acquisition times. A key development will be the ability to identify sparsely distributed targets by adapting targeting algorithms developed for defense and remote sensing. Combined, these techniques will allow us to test the limits of lowering the incubation period and transition from isolated cells to food matrix environments. However, even with the techniques developed in our studies, identifying pathogens is still a difficult and time-consuming process. So we are developing methods to increase sample loading and shorten time to answer. This includes innovations in sample loading technologies such as microfluidics to draw samples directly from incubation or even the line for analysis. Targeting algorithms combined with a large field of view collection optic can be used to rapidly identify areas of interest with potential pathogen cells. The goal of our work is to develop a fully contained system with the elements of hyperspectral imaging, dark-field microscopy, machine learning, and automation to deliver results with low incubation periods and minimal preparation in a simple, rapid process that can be operated without significant training. Well, thank you again for listening. We're happy to answer any questions you might have.